for now.
Hello everyone, I'm really happy to see people joining us today to our first keynote lecture, Gene Regulation and Cancer by Skirmantas Kriaučionis, and it is my great honor to present him. Dr. Skirmantas Kriaučionis is an associate professor at the University of Oxford Ludwig Institute for Cancer Research. In 2012, Skirmantas Kriaučionis was honored and awarded by the Lithuanian Ministry of Education and Science for discoveries in the field of DNA research and collaboration with Lithuanian biotechnologists. With his research team, the doctor explores the molecular function of DNA modifications in normal and cancer cells and aims to reveal the principles of how DNA modifications affect transcription and identify keynotes for epigenetic intervention. Thank you, lecturer, for joining our conference. Without further ado, please welcome Dr. Skirmantas Kriaučionis.
Hello, thank you for a very kind introduction. I must apologize, my webcam has somehow decided to divide screen in two parts, so I may suddenly appear twice. Okay, so let me try to share the screen and present you with some of our recent work. I hope you can see the presentation. If not, please let me know. Again, thank you very much, organizers, for invitation. It's always a great pleasure to talk to students and to share some of the insights into understanding how gene regulation works in general and how perturbation in gene regulation are impacting cancer. So one of the interesting paradigms and one of the probably most interesting aspects of human development and development of many multicellular organisms is the fact that we start as a single cell, and in this image you can see the fertilized oocyte, and the single cell is capable of producing many different cell types which operate in us and can perform very different functions. And this one genome is capable of producing these diverse functional cell types and units. And what is interesting, that's how one genome is capable of achieving so different morphological and functional manifestations is the fact that in each cell type, different repertoire of genes is being employed. And interestingly enough, most of the cell types still preserve that part of the genome which is called non-use genes, which is not required for function of the cell type. For example, neuronal cell types, they still have genes in their genome which are important for function of liver or any other organ. There are one exception probably to this, we know erythrocytes. The final stages of development, they do remove nuclei from cells and they end up being not having at all genomic DNA, which resides in the nucleus. So how it is being achieved is that genes are turned on and off at specific cell types. And this was the principle which was discovered by Jacob and Monod, is that DNA, which is depicted here as double helix, and gene, start of which is indicated by an arrow here, can be turned on and off by the property of proteins which are called transcription factors. Transcription factors are proteins which are capable of recognizing specific sequences of DNA. And by doing so, they're able in turn recruit many other additional proteins, such as in this case RNA polymerase, which would be able to turn this gene on if it is required to operate in a specific cell type. And we know that transcription factors are critical and essential for this, and this is the principle which was established and awarded Nobel Prize work by Shinya Yamanaka which was awarded Nobel Prize more than a decade ago, shows that if one takes a cell type which have its own identity, for example, a fibroblast, and introduces a certain set of transcription factors, these 4 transcription factors in this example, these transcription factors are capable of changing identity of the cell from the fibroblast to stem cell. So this process is called reprogramming. It is slow, it takes weeks, and it's very inefficient, but it shows that if right transcription factors are present in the cell, they're able to execute a program of genes which would be able to change cell types. So sometimes development doesn't go in the right direction, and what is called adversarial. Cellular organisms in humans as well, normal tissues can transform and start gaining phenotypes which are not necessarily beneficial for the function of the whole organism. And this is cancerogenesis and transformation into a diseased state called cancer. And sometimes it is multi-step process where there are less malignant states of cells transforming and evolving into more malignant states, which end up with very severe and debilitating disease, cancer. And just to show a few examples, for example, normal tissue esophagus can gain state which is called Barrett's esophagus. And Barrett's esophagus has higher probability of transforming into esophageal adenocarcinoma, which is a very deadly cancer. And as well, as another example, in normal blood, it can develop phenotypes which are called myelodysplastic syndromes. And again, these are precancerous conditions which lead often to higher chances of acquiring, again, a debilitating disease called leukemia. Although it's not necessary that in every cancer there is this intermediate, less malignant state. There are instances where either these states are not observed or they don't exist, and some more pronounced oncogenic events are transforming normal cells directly into cancer. So there is this similarity, what I mentioned, between normal development, reprogramming, and transformation in tumors. The perhaps most substantial difference that the driving force for normal cells to become cancer cells are mutations. So mutations which appear in the DNA are able then to change the phenotype through the events, sometimes which take quite long time and involve multiple mutations. They take to events which lead to the formation of the tumor. This is similar to what I explained what happens in reprogramming when we can introduce the transcript factors which would be able to change phenotype of the cell. So this process of transformation is thought to be not necessarily instant. It's always happening when cells divide, and normal cells may receive DNA damage from aging, inflammation, or reactive oxygen species, which would lead into certain mutations and perhaps epigenetic instability, which would result in the producing of tumor cell, which later on is able to multiply, and each multiplication would produce some other subclones which would have certain features which could be selected for and start change the tumor composition. And perhaps the most challenging aspect in tumor biology is the fact that tumors are composed of these variety of cell types, which are driven both by different mutations and genomic instability, because tumor cells typically don't have such a high fidelity replication as normal cells. But as well, it's driven by the fact that tumor would be having different challenges, and by growing, for example, tumor would develop zones which would have less access to the nutrients, less access to oxygen, and which would start executing gene expression programs which are related to hypoxia or to starvation. And this really heterogeneous tumor makes it's a very challenging way to try to tackle treatment of it. So again, it's now very well established that cancer is not one disease, it's multiple very different diseases. And in different tissues, we know that genes which are being mutated are often very different. You can have even different genes mutated in the cancers which are originating from the same tissue. But in addition to that, there is some specific patterns. The exception to this actually is this tumor protein p53, which, as you can see, is mutated in many different cancer types. So
this is one of these. Interruption.
Yes.
You can see only the first slide.
Oh no. Okay, that's strange. Let me solve that. Right. Let me try to reshare, perhaps stop sharing and reshare, and let me know if it helps or not. Maybe I'll share the whole screen this time. Let me, I'll maximize the screen and try changing the slides. Tell me if you see slides changing.
Yes, now we can see it.
Right. I'm sorry for this. I guess I'll still probably continue where I stopped because I don't want to repeat and what I already said. Okay, so what I wanted to mention in this slide is the fact that many often in different tissues, we identify a different set of genes which are being mutated, with the exception of this tumor protein p53, which is one of the most common across different cancer types originating in different tissues, mutated gene. But for example, if we look at some other genes like IDH2, it's very infrequently mutated in breast cancers or bladder cancers or DNMT3A, and it's more frequently mutated in acute myelogenous leukemia. So each tissue has its own set of cancer-driving genes which would lead to transformation. So I would like now, after giving you this general introduction, to focus my talk on two subjects which we work on in my group. So first, more generic questions: how does DNA mutate? I explained to you that mutations in DNA are the key events leading to transformation in cancer. So we want to understand how and what affects mutability of the DNA. And second, how does these mutations cause behavior differences in cells? So what is this transformation process leading from mutation into a cell which have very different set of phenotypic features which are detrimental to the normal function of an organism? So to split these two broad questions, the second question we try to understand is in more detail how transcription genes is regulated. Since, as I told you in the beginning of my talk, transcription of the genes, differential transcription of the genes underpin different cell phenotypes. So before I start talking in detail about these two aspects of our work, I would like just to remind you one aspect of the DNA which we are very much interested in. These are DNA modifications. And as you know, DNA is composed of 4 bases, GATC, and one base, cytosine, which structure is shown here. It's a rather unusual base because it's capable to receive DNA modifications. So modifications which it receives is methylation, which is modifications. So there's methyl group attached to this 5' position. making 5-methylcytosine. And 5-methylcytosine is in typical human genome would be around 48% of all bases. Slightly less frequent modification of 5-methylcytosine is 5-hydroxymethylcytosine, which is around 0.8 to 0.1% of bases in the genome. And there are other two modifications which are very rare in the genome, and they are thought to be demethylation intermediates. These are 4-mil and carboxyl cytosine. So my talk will focus on these two modifications, 5-methyl and 5-hydroxymethylcytosine. So these modifications interestingly reside in context neighboring G. So CG, the nucleotides are most frequently methylated or hydroxymethylated. And methylation is being deposited by 3 different enzymes of DNMT family. And then methylcytosine becomes substrate for hydroxylation, and hydroxylation is executed by that family of enzymes. And hydroxymethylcytosine can be further oxidized to 4-mil carboxyl cytosine, which can then be removed from DNA, causing unmethylated cytosine. So this enables erasing of 5-methylcytosine or 5-hydroxymethylcytosine marks in the genome. These different states of modified cytosines are recognized by different proteins. So CXXE domain is a common module among chromatin proteins which bind to unmethylated CGs, whilst MBD proteins bind to methylated CGs, and there are some genes which have been attributed to recognize 5-hydroxymethylcytosine in the genome. So as I told you, these DNA modifications are important for normal development. If DNA methyltransferases or all that enzymes are being knocked out cells, these would not be able to produce normal viable embryos. Important in brain development, because MECP2 gene is frequently mutated in neurological disorder Rett syndrome. Important normal hematopoiesis, and we know that because in acute myelogenous leukemia we often observe DNMT3A or TET2 mutations, and these mutations typically lead into cancer, so it's important in cancerogenesis as well. So how DNA methylation is propagated when cells replicate? This is important because it constitutes DNA methylation as epigenetic signal. So when DNA replicates, DNA polymerases create their DNA strand which doesn't have DNA modifications. In this picture, I depict 5-methyl CGs as these dark lollipops and unmodified CGs as light lollipops. And after replication, we get unmodified bases being incorporated, but there is this DNMT1 UHRF1 complex which interacts with pCNA and travels with replication fork, which would be able to re-establish methylation pattern on the daughter strand, the same way as is found in the parental strand. So this is actually a really beautiful mechanism how DNA methylation is being inherited during the cell division. So as I told, DNA methyltransferases create 5-methylcytosine in the genome, and these are found in the CpG, the nucleotide context. If we look across the genome, most of the genome is actually highly methylated. This is methylation frequency distribution across the whole genome in humans. But the small proportion of the genome have sites which are unmethylated. And this is how typical genomic locus would look like. Again, I represent methylation as lollipops, and we have these regions which are termed CpG islands, which have unmethylated CpGs, whereas the rest of the genome is highly methylated. Other sequences which are called enhancers, which act as well as important regions to help to turn on and off genes, we are as well found in a hypomethylated state. Sometimes these regions can get methylated, and we observe this often in tumor suppressor genes in cancer cells. So they acquire methylation of the CpG island, which is correlated with silenced activity of the gene. Some testis-expressed genes are methylated to normal tissues, and this is the mechanism which ensures that these genes are not expressed in other cell types. Imprinted genes would have one of the alleles methylated, which enables different control of two alleles in the same nucleus. And typically we see methylated CpG islands on genes on inactive X chromosome in females. The heavily methylated part of the genome encodes retrotransposons and elements. So now, after introducing 5-methyl and 5-hydroxymethyl cytosine, I will tell you about one of observations we had, which illuminates how cytosine bases with different DNA modifications mutate. So how does DNA mutate? So typically, DNA may receive some external mutagen, and this will be a photon coming from UV light, which can induce crosslinks, which would cause what is called DNA damage. And if this DNA damage is then unrepaired, it can produce mutations. Another source of mutations is more related to intrinsic errors and biological events. So not necessarily one would need to have exposure to external mutagen, but infidel enzymes which are involved in either replicating or repairing DNA can cause appearance of the mutations. So most mutations, actually nearly all mutation types, are linked to DNA replication. And the reason for that is because if DNA is damaged and there is some unusual structure in the DNA, this can still be repaired. But the problem happens if these cells replicate, and somehow, if this damage is not repaired and DNA polymerase is able to replicate it, then it produces mutation. And that mutation is then looks like normal DNA to cells and cannot be reverted or repaired. So often we see that DNA replication is one of the important mechanisms which would produce from damaged DNA which can contain mutations. What kind of mutations can happen? So there are 3 types which are depicted here. One can have substitution mutation, where one base in the genome is substituted to other base, and here we see C2T mutation. We have insertions, where some sequences inserted, or deletion, which some sequences are deleted. So my next part of my talk will mainly focus on substitutions. So when we look at the substitution and group by type of substitution which can happen, and this is an image produced in our group, figure produced in our group when we looked at mutation types which happen in brain cancer, and we group them by type of substitution, so C2A, C2G, C2T, and then we group by nuclear contexts which are either 3' or 5' from the base which is being mutated. So if we look at this distribution of mutations in brain cancers, we see that there are certain types of mutations which are very frequent compared to the others. The C2T mutations are the most frequent mutation type, indicated by this bars here. And not every C2T mutation, but especially C2T mutation where C resides in a nucleotide composition with CG is the one which have increased mutation frequency. And this is actually observed not only in brain cancers, this is observed across multiple different cancers with C2T mutations within CG containing the nucleotides more frequent than other types of mutations. Why is that happening? It's thought to happen because C within CG nucleotide can be methylated, and this methyl CG has higher probability of losing this amino group. And this is a spontaneous reaction which happens due to interaction with water. This deamination reaction produces a C2T mutation. But this is already known, but in this study we asked what happens to 5-hydroxymethylcytosine. What are the effects of this base on non-mutational rates in cells? So to do that, and this is purely computational project. In this particular project, we have not done many experiments ourselves, but we used already published various ways data. So we used a map of DNA modifications 5-methyl and 5-hydroxymethylcytosine, and we try to correlate it in various mathematical ways with patterns of mutations which are observed in brain tumors. We chose brain because brain is tissue which have most hydroxymethylcytosine when compared to other tissues. And we try to correlate the two. In here we plot just chromosome 3, and we plot 5-methylcytosine frequency and C2T mutation frequency in CpG denucleotides. And we observed that there is a very good positive relationship. So places which would contain higher 5-methylcytosine frequency, they would display higher C2T mutations. When we plotted 5-hydroxymethylcytosine distribution across the same chromosome, we observed that actually there is an inverse correlation. The sites which contain high hydroxymethylcytosine frequently would often have reduced C2T mutations, and this R value indicates the reverse relationship between 5-hydroxymethylcytosine abundance and mutation frequency in CpG nucleotides. So we can plot this information for the whole genome in this kind of plot. So I'll go through it. In here, we bin the genome in different groups depending on how heavily modified it is. So this particular groups would have from 0 to 20% cytosine being methylated, and this would be very heavily methylated cytosine. And when we grouped according to methylation frequency, and the color in here indicates the mutation frequency, we observed that with increased methylation, we observed increased likelihood of mutation, C2T mutation. So now we asked what happens when methyl cytosine is hydroxymethylated. So if we go across this diagonal, we can observe that then mutation frequency drops. So we observed that actually 5-hydroxymethylcytosine containing sites, they have similar mutation frequency to unmodified cytosine, but substantially lower mutation frequency than methyl cytosine. So this was done by using observation in brain, and I told you this is a density distribution of hydroxymethylation values. The brain contains highly hydroxymethylated genomes, whereas, for example, kidney and blood contains lower frequency of 5-hydroxymethylcytosine. But we still have some sites which are hydroxymethylated. So we took those 5-hydroxymethylcytosine high sites, and we observed that they have lower mutation frequency than sites which contain high frequency of methylcytosine. So this is consistent between different tissues, that 5-methylcytosine is more mutagenic and 5-hydroxymethylcytosine is less. So we can then take and try even in unbiased fashion to relate using generalized linear modeling different distribution of 5-hydroxymethylcytosines with mutation frequencies, and this unbiasedly recapitulates the relationship that brain hydroxymethylcytosine actually relates to brain C2T mutations. So what is the biochemical basis for this? So as I told you, one way cytosines mutate is by losing this amino group. if a phytogen loses amino group, it produces uracil, which is a not natural DNA base. If 5-methylcytosine deaminates, it produces thymidine. And this reaction is actually not equivalent for unmodified and methylated cytosine, and this is published by Thomas Lindhoff in the 70s, and we nicely recapitulated this observation in our hands, showing that deamination frequency in vitro in water of 5-methylcytosine is higher than unmodified cytosine. So we asked them what's deamination frequency of 5-hydroxymethylcytosine, and we observed that 5-hydroxymethylcytosine deaminates at similar rate to unmodified cytosine. And this could be one of the molecular mechanisms by which 5-hydroxymethylcytosine is less than 5-methylcytosine. But then an interesting observation we had when we looked at tumors which have especially high number of mutations. So this is a Cancer Genome Atlas network effort to produce and aggregate data across different tumors, looking at how many mutations are being observed in different tumors. So typically, most of the tumors would have approximately 1 mutation per megabase, but there are some tumors which accumulate very high number of mutations. So these tumors, when you look at what specific status they often have mutations or inactivation in genes which are involved in mismatch repair, or even mutations are being observed in enzymes involved in replicating DNA. So this is polymerase epsilon. It's a major DNA replicating enzyme in cells. So these mutated tumors, they gain a lot of mutations, and we asked what type of mutations are being observed, whether we observe increased frequency of CTT mutations in CpG denucleotides. And surprisingly, we actually did observe that there is high number of CTT mutations in CpGs in cancers which have polymutation and microsatellite instability. Why would that be happening? And we then looked at it orthogonally, trying to relate mutation frequency with replication origin. And we observed that if we aggregate all mutations along replication origins, and you look at tumors with polymutation, that there is enrichment of mutations in the leading replicating strand. And poly is known to replicate leading strand. So this suggests that polymerase epsilon has some defects perhaps when replicating cytosines containing methylated cytosine. So what could explain these results? So one possibility would be that polymerase epsilon and mismatch repair is involved in non-canonical repair of mismatches generated by spontaneous deamination. But in this case, it will take a very long time to accumulate number of mutations which are typically observed in tumors with these samples. And another possibility that these produce gain-of-function mutation phenotype, which would then increase mutability at 5-methylcytosines. But we observe some effect even in cancers without mutations of polymerase epsilon and MSI phenotype. Therefore, probably this is not the likely possibility. So the hypothesis which we are working now on is that polymerase epsilon have low fidelity while incorporating nucleotides opposite 5-methylcytosine. And we developed some in vitro assays which would enable us to measure what error rates are of polymerase epsilon when replicating different modified bases containing templates. So this time I would like to switch gears and go a little bit more into gene regulation. So how mutations cause changes in cell behavior, and in more detail, how transcription of genes is regulated. So before I start talking about that, I would just like to remind how DNA is being organized in cell. So this is electron transmission electron microscopy of Purkinje cell nucleus. So that's where genomic DNA resides. And it resides at the atomical level in close complex with histone proteins, which makes this fundamental unit called nucleosome. So this is histone octamer, and DNA is surrounding nearly 2 times this histone octamer. And this is then in less organized fashion packed into the nucleus and form this chromatin fibers. So chromatin is an important aspect of gene regulation, and histones especially are playing important roles in it. And we know that from a couple of reasons. And one of them that these histone proteins, which form nucleosome, they are heavily modified and split at their tails and terminal tails. So almost every residue is important in depositing these modifications. And modifications can be methylation of arginines, acetylation of lysines, phosphorylation, and these type of modifications correlate very well with activity or functional properties of these DNA sequences where they reside. So the principle by which these modifications they are deposited, written, and read are by different enzymes. So for example, acetylation of lysine is being written by histone acetyltransferases, which would produce acetyllysine. Histone deacetylases remove acetyl mark, and other group of proteins like bromodomain proteins are able of recognizing this mark and helping to manifest into the function of that particular chromatin. So chromatin is as you saw, this tracking of the DNA across the nucleosome, and this is very schematic representation of each nucleosome and DNA surrounding it. It becomes kind of barrier for transcription. And if we take this example in here, we know that genes would have low activity or no activity, which are not being expressed, typically would have methylated promoters, which would be depicted by these black dots here, and would have specific as well histone modification patterns. But when these genes are being activated, go from inactive to active state, transcription factors are being recruited, and typically genes would be unmethylated and specific histone marks deposited here, which would help to maintain active state of a gene. And this would then enable recruitment of many other proteins, which would ensure that RNA polymerase launches into productive transcription and it's able to produce transcript and ensure that the protein product is being available in cells. So we're interested to know how transcription factors interact with this chromatin to enable transcription. We specifically focused on 2 things, on 5-methyl cytosine and histone acetylation. So one way 5-methyl cytosine impacts on gene regulation is that it can affect interaction with some transcription factors. For example, MYC and ETS transcription factors, they contain CpG recognition sequence. And if cytosines are methylated, they would have substantially lower affinity and would not be able to bind effectively at sites containing 5-methyl cytosine. Whereas other transcription factors can tolerate 5-methyl cytosine at recognition binding sites. And another mode how methyl cytosine works, it can have recruitment or repelling of some proteins which specifically interact with modified CpG nucleotides. So methylated CpGs attract methyl CpG binding proteins, and they would repel CXXC domain proteins, which are preferably interacting with unmodified CpGs in the genome. So because the way MBDs work, they are typically in complexes which contain histone deacetylases. And to interrogate the function of these complexes, we decided to block methylation and block histone deacetylase activity, and ask how does it reflect on transcription factor binding and function of the chromatin. To do this, we use two techniques, and I would like to quickly explain this. So one technique which is used to measure how transcription factors or other chromatin proteins bind to DNA is called chromatin IP. And chromatin IP is together with sequencing, it's a very powerful technique which enables to study DNA binding protein distribution across the whole genome. So the way it works that when we have DNA bound by some protein of interest, we expose these proteins to a cross-linking reagent, which would covalently bind the protein to the DNA. And then we would be able to shear this protein DNA complexes into smaller pieces, and we can use antibodies against proteins to enrich for proteins which have affinity to the antibody. In this case, we lose DNA together with protein DNA complexes which bound to any other proteins. And then we can extract these short enriched pieces of DNA bound to the protein of interest and sequence it using next generation sequencing. So we get the distribution of short fragments and we can align them across the genome. And typically at places where this protein is frequently bound at the DNA, we observe enrichment of these short DNA fragments. And then we can aggregate, and we receive typically plots like that, where we see peaks, in this case at the positions which contain particular histone mark, but the same is true for DNA binding proteins. Another technique which I would like to introduce you is assay for transposase accessible chromatin called ATAC-seq. So this assay measures regions of the genome which are accessible. And it works by exposing nuclei to transposase DNA complexes, which would be able to go to regions which are accessible and introduce these primers into them, which can be used then both to fragment and amplify DNA. And we would be able then to, again, enrich for these reads from accessible regions and sequence them to identify and map regions in the genome with high accessibility. So what we did, we had cells in which we had no DNA methylation, so all 3 DNA methyltransferases have been knocked out. And we treated these cells with HDAC inhibitor, and we asked how transcription factor binding and accessibility and gene expression change in these cells. So just to show that our treatment works is that when we treat with HDAC inhibitors, we observe increase in H3 acetylation in these cells. And then if we map H3 acetylation across the genome, we observe increased signal both in peaks which have to start with high acetylation signal, as well as elsewhere in the genome. So we see increase of the signal across the genome when we inhibit histone deacetylases. So how does it impact transcription factor binding? So we use this ATAC-seq approach and targeted approach, chromatin IP-based approach, which I explained before. And this is to show you first the ATAC profile. Again, we see peaks which indicate regions which are highly accessible in the genome. And if we compare what happens to accessibility, then we don't have DNA methylation. We see some regions which gain accessibility, some regions which lose accessibility. And this is one example of a region in the genome which gains accessibility when DNA methylation is absent. And then we try to examine what is the relationship between mean methylation state and the ability to lose or gain DNA methylation. And we observe that those regions which gain accessibility, they tend to have highly methylated regions to start with, as it's shown in this example. So by looking at accessibility patterns, we could then look for what transcription factor binding motifs, these newly accessible regions are enriched in, and we found that regions which are enriched in sequences, regions which are gaining accessibility after loss of DNA methylation, are enriched in transcription factor binding sites of NRF1, NFY, and EBOX classes. In addition to looking across the genome, we observed that there is increase in accessibility, which is observed in regions which are called rep-mask regions, so which represent repetitive portion of the genome. And this was especially pronounced when we treat cells with HDAC inhibitors, with TSA. So what are these repeats? So our genome is composed, actually nearly half of the genome contain these repeated sequences, which are of either retroviral or transposon origin, or some simple repeats. And these regions are typically highly methylated. And often in cancers, they are being found that they are hypomethylated. And this causes increased activity of various retrotransposons, which can induce genome instability. So when we looked in our cells, when we treat cells with histone deacetylase inhibitor, we observed that there is an increase of accessibility in line and LTR type of repeats in the genome. And typically the younger repeats would have higher increase in accessibility than older repeats. So then we employed a targeted approach. We mapped 5 transcription factors and we asked how binding of these transcription factors change in the genome where we alter DNA methylation or histone acetylation. And we observed various scenarios. So, example, transcription factor in here, gaining binding sites when DNA methylation is being lacking. And in this case, it responds to histone deacetylase inhibitor, so it gains binding in this particular site when we treat cells with histone deacetylase inhibitor. In some other cases, we observed that there is gain of binding only in the sites in cells which we treat with, which are deficient of DNA methylation, are being treated with HDAC inhibitor. So this is for GAPDA, MAX, and YY1 transcription factors. So in these cases, DNA methylation and HDACs, they collaborate to restrict binding of transcription factors. So when we look across many different sites, we observe that different transcription factors have quite a distinct ability to sense chromatin and DNA methylation effects. So for example, NRF1 is very sensitive to loss of DNA methylation. So we see that sites are being gained in cells which don't have DNA methylation, but very few sites which alter binding when we treat cells with histone deacetylases. SP1 transcription factor is actually not very sensitive at all to either single loss of DNA methylation or with inhibited HDACs, but there is more sites being gained when both of these events are being administered. When inhibit HDACs in cells which don't have DNA methylation. And MAX, GAPDA, and YY1 again show much more pronounced effect when we inhibit DNA, inhibit histone deacetylases in cells which don't have DNA methylation. So this suggests that DNA methylation and HDACs act as probably independent mechanisms to maintain some chromatin sites unbound. So when we look at where these binding events are happening, we observed again that for YY1 transcription factor and for GAPDA transcription factor, there is substantial proportion of sites which are being residing in the repeated part of the genome in line, LTR, and sign elements. And in this, we try to relate both accessibility, transcription factor binding, and gene expression. And the first thing that we observed that GAP-BA binding and YYY binding, which is related to increased accessibility, can as well cause elevated expression of these transcription factors, of these repeat elements. And this is typically pronounced for this L1 type of the repeat in the genome. So in summary, what we observed by studying how transcription factors interact with chromatin containing different modifications is that line and LPI repeats of the genome are robustly silenced with mechanisms that include both DNA methylation and HDAC activity, both of which is important in restricting transcription factor occupancy, which was mostly pronounced for YY1 and GAP-BA transcription factor. But DNA methylation and histone deacetylation are largely independent mechanisms of silencing. And the role of DNA methylation restricting transcription factor binding is more common, as inferred from previously from accessibility change experiments. And we see that whereas DNA methylation has a limited role facilitating transcription binding in vivo. Okay, so at this point I would like to stop my talk and acknowledge people who did most of the work. So the first part, which was investigation of relationship between DNA modifications and mutability, was done by talented computational biologist Marketa Tonkova, and the transcription factor binding in different chromatin contexts was done by Martin Cusack. They both now left the lab and doing their postdocs. And thank you very much for attention.
Thank you for the interesting and very important presentation and for being a part of our conference.
Thank you for the invitation.
First day of International Health Sciences Conference 2021 is over. I hope you all enjoyed it, and tomorrow we will meet for more scientific sessions, workshops, and our second keynote lecture. Thank you and goodbye.